thanks very much for inviting me and getting a chance to speak here. I think it's a wonderful initiative. And then also congratulations to the organisers. There's been some fantastic talks over the last kind of couple of weeks. So I've got some big shoes to fill, but hopefully I can do this area justice. So as Craig mentioned, I'm from the Health Products Regulatory Authority of Ireland. And my talk is really going to be kind of focusing on how does a medicine get a license? So how does it get a license to sell its product in Ireland? So Finbar talked very nicely about like kind of drug discovery and so on. So how do we go from that discovery of a promising new compound to it ultimately getting a license that can then be sold in Ireland? So whereas Finbar is very much at the coalface of the discovery, we're much more towards the end of the process where we kind of evaluate all of the evidence and then ultimately grant the license. But I'll talk about that a little bit more on that process in terms of how we do that now throughout the talk. But Craig very nicely introduced the Health Products Regulatory Authority of Ireland. So we essentially, in a nutshell, regulate health products. So we have this responsibility to ensure that different health products in the Irish market demonstrate their safety, their efficacy, and then also that they're produced to a really high standard. So there's regulatory documents that outline the very specific standards that different health products need to meet to demonstrate their safety, their effectiveness, and so on. So we regulate health products. So we have a responsibility for ensuring that different health products are maintaining the standards required to ensure that they are safe and effective. So in terms of, we regulate medical devices, blood, tissues, cells, and organs, and Finbar touched on that, certain types of controlled drugs. We also ensure, again, that cosmetics maintain kind of high standards. But then we also regulate different types of activities. So we regulate clinical trials. So we ensure that the practice of clinical trials are attaining the high standards and prioritizing patient safety. But then we also regulate the actions of the use of animals in scientific research as well. So again, looking to ensure that the use of animals that the facilities using animals in research are kind of maintaining really high standards in terms of their practice. But for today's talk, I'm really going to focus on the HPRA's role in that process of granting a license to a new medicine. So what is it that we do and what the companies have to do to demonstrate that they have enough evidence to say we deserve a license and we're going to sell our product on the Irish market. So how does a drug get a license? So it's a very long process. So from that stage, from that early discovery phase that kind of Finbar was discussing, the identification of a new microRNA, for example, that shows a really promising effect in maybe cancer cells in a lab, to ultimately the generation or the development of a medicine. It could be a pill that ultimately a patient takes at the end of the day to treat their illness or disease. It's a very, very long process. But essentially what the company has to do to get a license, they have to provide three main types of evidence. And the three main types of evidence that companies need to generate over the course of this life cycle, I suppose, from going from your basic discovery to a medicine, the company needs to produce data pertaining to the pharmaceutical quality. So I'll talk about these in a little bit more detail now in a second. So the first package of data that the company needs to produce relates to the pharmaceutical quality. They need to produce evidence in non-clinical models. Again, I'll talk about this in a second. And then the last big package of data that a company needs to produce in order to get a license is our clinical trials, so information derived from clinical trials. And the whole point of these three different packages of evidence that a company needs to produce is they're trying to demonstrate that this new medicine is safe, that it works, and they're able to produce it to a really high standard. So that's really the essence and the core of these different types of evidence that the company needs to produce. And then ultimately, that's then submitted to a regulatory body like the HPRA or the EMA, and then all of that evidence is then assessed. So a bit of a spoiler alert kind of coming up. So the key take-home message from this talk and from the next kind of few minutes is that our job as a health products regulatory authority is to assess all of the data that a company has generated and to actually make sure that the evidence does suggest that the medicine is safe, that it actually works, and it is manufactured to a high standard. So we make sure that any claims that company are making in relation to these kind of elements is actually backed up by data. So that's our primary role in this process. So I'm just going to give you a little bit of an insight into the types of evidence that the companies need to submit to support their application for a license. So the first part of that, I suppose, trio is, can they make or can they produce the medicine to a really high standard? And this comes in their pharmaceutical quality package. So this big chunk of evidence that they have to submit to regulators. And Finbar, again, nicely kind of set the scene there. So in terms of some companies or some medicines might use cells to produce a medicinal product, or it might be a chemical process that takes many different steps and to produce over time the final product. But during each step in the manufacturing process, so however many steps that takes, byproducts or waste products can be, and very often, produced in that process in terms of the manufacturing of the final medicinal product. So the company has to submit a big package of pharmaceutical quality evidence, and essentially what they have to do is show a really detailed plan and understanding of, first of all, how we produce the medicine. Is it using a cell system? Is it a basic chemical kind of process? So how is the medicine produced? What waste products are produced during this product or process? How do they control the amount of waste products they are producing during the process? So are they implementing processes to reduce the amount of waste products that are produced? How are they removing the waste products? And then again, it's this batch to batch consistency. So it's okay. It's all well and good making one batch of a really pure and potent medicine, but can they do it time and time and time again? So that's a really important part of this type of evidence that the company needs to produce. So is it a pure, is it a potent substance? Can they do it time and time again? And then we also perform inspections to make sure that the data that's actually being submitted to outline all of these processes is actually being done. So we'd have inspectors that go to the manufacturing facility, look at the process, look at all the information, and again, to make sure what they're actually telling and submitting is actually what's happening on the ground. And then finally, a company, because this is a really controlled process, this manufacturing of a drug, it's really controlled. So a company can't just change it or can't change a step. So they're legally obliged to notify a regulatory body, such as the Health Products Regulatory Authority, if they're changing any steps in this process, and they have to provide evidence and data to support that. So that's the first batch of data that a company needs to produce. Can they manufacture this to a really high standard? The next set of data or evidence that a company has to provide comes in the form of non-clinical evidence. So these are really some drugs, depending on the approach I get. Some drugs can be really experimental. So back to Finbar's lab, like this kind of drug discovery of new compounds or new chemical entities. So before we ever test this in a human, it has to go into preclinical assessment. So essentially what we want to do, or the purpose of this preclinical assessment, is to build up a safety profile, to get an understanding of, first and foremost, the impact of the drug on the body. So again, looking down at a cell in a dish, and this compound has great potential to maybe reduce the size of a tumor or whatever the outcome might be. They need to demonstrate this proof of principle in an animal model. So they might take an animal, a cancer model or a mouse model of cancer, give the drug and see whether it has an effect. Another kind of key aspect of this impact of the drug in the body is to understand the safety profile. Does it have an effect on the central nervous system, the respiratory system, or the reproductive system, for example? So again, it's really generating that picture of what impact does the drug have on the body, but then also it's just get an understanding of what impact the body has on the drug. So when you take it, how is it absorbed? Where does it go in the body? How is it broken down? And ultimately, how is it excreted? So animal models, or preclinical models, are really important for this. Another really important aspect of the preclinical assessment, so again, before we ever test this in humans, is to try and identify what dose do we give. So this is a really nice quote here. So poison is in everything, and in nothing is without poison. So it's the dose that really kind of determines the effect. So again, if we have a mouse model of cancer and we have this really promising kind of drug, if we give it in too low a dose, there's no effect, and ultimately we have an unhappy mouse. On the flip side, if we give too much, we might poison the mouse, and again, we've induced this toxic state. So ideally, we want to identify what's the sweet spot, what is the therapeutic effect where we can maximize the effect of the drug but minimize the side effects. So the preclinical testing phase is a really important aspect in this development of a drug, and it helps to inform what dose is then ultimately used in humans, which is the final, I suppose, batch of data that the company needs to produce to demonstrate again that they can produce this drug to a really high standard, and then also understand the safety and the effect of the drug. So is it working? So it's the approach to clinical trials is a very progressive approach. So we've gone from that very experimental discovery in the lab. We've tested it in animal models. We have a good idea of the safety profile, but we now start to test this in human models. So give it to individuals to see what, first and foremost, is it safe? What are the side effects? And then ultimately, is it having an effect? So this is a progressive approach. So we have a phase 1 trials, which the primary goal or the focus of a phase 1 trial is really to identify the safety profile, and it's typically given to healthy participants in small numbers, again, because we're just still building up this picture of, is it safe? We then, once we've determined that the safety profile of the drug, well, a good idea of the safety profile in a small number of healthy participants, and we see how the body is responding to the drug, but more importantly, how the drug is responding to the body, we then progress to phase 2 clinical trials, where we're still very much focused on safety, but now we're starting to see, okay, does the drug have an effect? So now we're starting to include patients in the phase 2 trial. So again, relatively small numbers of participants in the trial, but we're checking the safety, but also, is it starting to have a therapeutic effect? And there's also a dose finding element here as well. So we're still at these stages, we're still not sure what the optimal dose is. So there could be a couple of different dosing strategies used here. And then ultimately, again, if the drug, following these 2 phases of kind of investigation, is it safe? Is it starting to have a therapeutic effect? We then expand the clinical trials. And this could be, depending on the drug and depending on the type of disease, this could involve a couple of hundred to thousands of patients. And as we saw in the COVID vaccine trials, there was 40, 50, 60,000 participants in those trials. So really, really big trials. And again, constantly assessing the safety profile of the drug. So safety is paramount all the way through this. But now we're starting to see whether the drug has an effect or an impact on the outcome that we're interested in. So if it's a cancer drug, for example, is it having a therapeutic effect? But also we're investigating it under more real world scenarios, where bigger numbers of patients, maybe taking different types of drugs. So really investigating what it's like in a more real world scenario. as opposed to the smaller, more controlled phase 2 clinical trials, and then in the phase 1 trials. So once a company has generated all of this evidence, so their pharmaceutical quality data, can they manufacture the drug to a high standard? What is the safety profile in animals? What's the pharmacokinetics? What's the pharmacodynamics? So what's the effect of the drug on the body? What's the effect of the body on the drug? And is it having a therapeutic effect? So once they've generated all of these types of evidence under these three headings, it's then submitted to a regulatory body. And we ask ourselves some very simple questions. Does the data suggest that the drug is safe? Does the drug work, and how well? And is it manufactured to a very high standard? And then when we assess the data from all of these different packages, we then ultimately make that kind of question: do the benefits outweigh the risks? Because every medicine has a risk, every medicine has a side effect, but do the benefits of that medicine outweigh any of the potential risks? And that really informs our decision at the end of the day as to whether a company is granted a license to produce. So just to really kind of to drive home the role of the regulatory piece in this, kind of going from your basic drug discovery to the development of a drug that's given to a patient at the end of the day, our role is to assess all of the data, all of the evidence that's generated by the company. And our role is to really make sure that the medicine is safe, does it work, and is it made to a really high standard? And with that, I can see I've just gone over the 15-minute mark. So I'll leave it there, Craig, and hand it back over to you.